Hey everybody, I want to record a little video here about some of the electrical activities of the heart. Some of the story is going to be familiar from what you'll remember about how skeletal muscle functions, but there are a couple of tweaks and specializations that we're going to need to be aware of when we're looking at cardiac muscle. Now, you've at least attempted to see the cardiac muscle slides in the laboratory. Just some important things to remember along the way is that these are, again, striated muscles. They're striated muscles, and one of the things that you get from that is that really strong, fast, and efficient contraction, like what we used to see with what we were used to seeing with skeletal muscle fibers. Usually one of the most distinctive attributes of the cardiac muscle fibers are these structures that are called the intercalated discs. And what they have at each of these discs are both desmosomes that are going to be anchoring the two adjacent cells together, as well as gap junctions. This particular cartoon doesn't really show the gap junctions in the intercalated disc, but I can assure you that's where they're actually located in life. It kind of lines up the intercalated disc basically with where the Z disc would be on, the Z line would be at the end of a sarcomere. I'm not going to get into any more microstructure than that, but the thing to take home from this attribute of having gap junctions in cardiac muscle fibers is that these cells are electrically connected to one another. So an electrical impulse that is going to cause contraction in one cell will get passed from cell to cell so that the heart can act as what they call a syncytium, which is a bunch of linked cells that act as a unit. Now, the trace of a cardiac muscle action potential is different from what we've seen from looking at skeletal muscle fibers. First of all, orient yourself on this graph on the left here. So you notice we have time going from our 0 point over to 300 milliseconds over here, and our membrane potential. So now we recall that this is going to be this is going to be polarized down here, and this will be depolarized. We're into the positive numbers. So the blue trace is what's showing the action potential. On the other hand, this purple trace is showing you tension, or it's basically showing you how shortening is happening in the cardiac muscle cell. So this is like a composite figure. We'll compare this to skeletal muscle fibers in just a second. Okay, so from being at rest, a typical cardiac muscle fiber depolarizes really fast through the opening of sodium channels. But then there's this long feature called a plateau in which slowly opening calcium channels will keep the action potential charge relatively high until the as the sodium channels are opening, as the potassium channels are opening, and then as more of them open, potassium is able to leave the cell and cause the charge to drop down. So the same basic rules as what we saw in a skeletal muscle fiber is observed here, where it's this influx of sodium that initiates the action potential. But here the action potential is kind of elongated through this plateau where calcium ions, another positively charged ion, as it goes into the cell, that's going to keep the charge up pretty high before it drops off again. And also notice, the skeletal muscle fiber has a long refractory period, has a long period of time during which you're not able to start a new action potential. Now, this is important because what we don't want from a cardiac muscle fiber is tetanus. You recall that tetanus is that like maximally sustained contraction that we see in skeletal muscle fibers. You don't want the heart, when it's pumping hard, to go into tetanus, because then the heart's no longer a pump, right? So this change in the profile, that shape of the trace, is to keep the muscle fiber so that it's not going muscle cell, so it's not going to be able to stop contracting. Now, if we compare this with what we remember from skeletal muscle fibers, take a second, just pause the recording and compare the differences between the cardiac muscle fiber and the skeletal muscle fiber. I'll give you a second. Okay, so now looking at this, notice the time scale is really different. Notice that when you look at the time scale of the action potential is different. And notice you have a more or less bell-shaped curve here of the contraction of cardiac muscle fibers, whereas you have a quick, for a twitch, you have a pretty quick period of contraction and then a long tail for relaxation. That kind of profile we really don't see with cardiac muscle fibers. So I guess the take-home message from this is that there are some of the same attributes, for example, sodium channels and potassium channels, but the addition of the calcium channels makes this distinctive profile of an action potential in cardiac muscle cells. Now, in addition to that kind of characteristic profile, there are some cardiac muscle cells that are specialized. Specialized not really to contract, but just to generate action potentials on kind of a clock. Now, the cells do this are called pacemaker cells. So you notice that there's no real flat resting potential. The membrane potential will slowly kind of creep up. Then once it passes threshold, it'll depolarize, through the influx of sodium, and then as well as the calcium, and then it'll decrease down again, get down so that it's polarized, then come back up again for the same reason. So it does this on a bit of a clock. Now, this is where it starts. The physiology of muscle fibers starts to overlap with the anatomy of the heart itself. So you'll notice that the heart has within it a so-called intrinsic, self-contained conductance system. And this is governed by the kind of rhythmic contractions of pacemaker cells. As it turns out, the pacemaker cells that have the shortest clock are down here in a structure that's called the sinoatrial node. So the SA node is what's responsible for setting the basic rhythm of the heart. I'll point out the different structures as it kind of goes along. Basically, the action potential starts here in the AV node, and these arrows represent the action potential getting spread across the atria. Now, it gets spread across the atria because, remember, these cells are electrically connected through gap junctions. So anyway, it washes across the atrium, including to another specialized patch of cells called the atrioventricular node. This is just superior to that fibrous skeleton of the heart, but the skeleton of the heart has the effect of causing some electrical impedance. So basically there's resistance, so that the action potential that starts here basically stops once it hits that plate of fibrous connective tissue. So the action potential washes across the atria, but one way that it goes is to transmit the impulse to this structure called the atrioventricular node, which then bullets the impulse down to the apex of the heart through first the atrioventricular bundle, then it splits into the two bundle branches, and then those bundle branches, when they get down to these very lowest fibers, here they call it the subendocardial conducting network. In the old days, we used to call them Purkinje fibers, which is something you still see sometimes in the lit, for example, right there. These Purkinje fibers are like those terminal branchings of these. You'll see that the papillary muscles of for the atrioventricular valves, chordae tendineae, those are going to get messages to contract relatively early in the cycle. Now, so again, just to go through this again, the action potential starts here at the SA node, washes across all the atria, but particularly goes down to the AV node. The AV node then bullets the impulse down to the apex of the heart so that the contraction can begin down here in the cone of the heart, not going straight from the top down to the bottom. Because remember, we want the blood to be able to change directions. The blood's going to go through the AV valves and then out by way of the out by way of the semilunar valves. So anyway, this is the big thing to get about this. When we talk about how the heart cycle works in the next module, we'll use this conductance system as how it is to kind of explain how it is different parts of the heart are going to contract. And I'll show a little bit more of that in a moment. Notice here, this is the stereotypical ECG wave that you've seen before, at least in kind of cartoon form or in the movies and stuff. We'll identify particular events of the cardiac cycle that go along with these electrical deflections here. So the first one that happens is this little hump here that we call P, then which represents the period of time when the atria are depolarizing. Now, the ventricles, when they depolarize, they have a much higher deflection, this so-called QRS complex, where it kind of bounces around. So R is where you're going to have like that height of ventricular depolarization. Then over here you can see another kind of little hump that comes up for ventricular repolarization. So you might be wondering, you see ventricular depolarization here and repolarization here. How come you see atrial depolarization here but no atrial repolarization? The reason why you don't is it's kind of masked by this big QRS wave and actually contributes to part of its height, right? So when we're dealing with this later, here's the kind of the basic pattern for how it is these electrical events match on to where the activity of the heart is actually happening. So on these pictures, notice that where the parts of the heart that are depolarizing are going to be shown in yellow, and the repolarizing parts of the heart are in red. So first, during the P wave, the atrial depolarization starts here at the SA node and then washes across the atrial myocardium. Then, by the time it gets to the flat part before the Q deflection, basically you see the depolarization and contraction of all of the atria. Now, and after that point, the atria are going to start to repolarize, again, kind of stuck within this QRS complex. But then also during this period of time, you see this shooting down to the apex of the heart, the contractile wave that also is reflected by QRS. Then eventually, from kind of washing away from the interventricular septum as well as the apex of the heart, the impulse kind of washes up the two sides of the heart, and then gradually it's going to start to repolarize itself again. And as it's resetting, that's where it's showing the T part of the ECG wave. Then eventually, by the time you get to the flat part, the rest of the cone of the heart is repolarizing. In case you're wondering why they start with P, it's actually has to do with back when the ECG was first measured. They used people, when they used like your Cartesian graph, like a regular kind of grid, they would always label the origin as O, not 0, but just O. So P would be the next letter if you're going from left to right, and then Q is the next, R is the next, etc. I always thought that it seemed kind of arbitrary because it totally is, but that's kind of where it comes from, in case you're wondering. So, having said that, remember that the sinoatrial node is responsible for setting up like the baseline rhythm of the heart, but it doesn't do this by itself. I think I mentioned when we did the autonomic nervous system last semester that the vagus nerve on the parasympathetic side is what's responsible for slowing down the heart, and we also know then that the sympathetic side will be increasing its rate of contraction. So what the ANS does is it sort of calibrates the baseline rate. If you could denerve the heart, separate it from any kind of ANS stimulation, your baseline rate, heart contraction rate's closer to 100 beats a minute, not the probably 60 beats a minute that's typical for like young, healthy people. So ordinarily, the vagus nerve will slow down the heart when you're at rest, but then through activity or excitement, the sympathetic division will let it get up to its baseline rhythm and then exceed it. You see that the branching of the sympathetic part of this is a little bit more extensive in the ventricle, because you want the ventricles to really increase their output in response to emergency conditions in the body. That's the big stuff for now. What comes next is a treatment of the heart cycle.